Welcome to Enzyme Mechanism Class 2. Today we are going to discuss the catalytic mechanism of ribonuclease A. In our previous class we have discussed the different catalytic mechanisms employed by enzymes, and ribonuclease A is a typical example for an enzyme which follows general acid-base catalysis. And bovine pancreatic ribonuclease A is a small protein. It consists of 124 amino acid, a single polypeptide digestive enzyme which hydrolyzes RNA to its component nucleotides. And you can see RNA here, and ribonuclease A cleaves RNA into its component nucleotides through the formation of a 2' 3' cyclic nucleotide, which is an intermediate formed during this reaction, and later it is hydrolyzed. Coming to the active site of the enzyme, in the first module you have studied in detail about active site mapping, different techniques which are employed for finding out the amino acid residues which are present in the catalytic site of an enzyme. Similarly, the pH rate profile of ribonuclease A, which showed a peak at around 6, indicating the presence of histidine, and chemical derivatization study used especially alkylation, and X-ray studies indicated the presence of histidine 12 and histidine 119 at the active site of ribonuclease A. This means that these two residues, histidine 12 and histidine 119, which contribute to the catalytic process. And you all know histidine, which is having a pK value 6, which is near neutrality at physiological pH, and histidine can act as a acid in the protonated condition, and in the deprotonated form it acts as base, and histidine can act both as an acid and a base. And acid catalysis and base catalysis, when takes place simultaneously, which is known as concerted general acid general base catalysis. And ribonuclease A is a typical example for an enzyme which follows concerted general acid base catalysis. And in addition to histidine 12 and histidine 119, there is one more residue, that is lysine 41, which is also involved in the catalytic process. Here lysine 41, which stabilizes the transition state by interacting with the phosphate residue. So these three residues are involved in catalysis: histidine 12, histidine 119 and lysine 41, and among these histidine 12 and histidine 119, these two are involved in acid base catalysis. And coming to the mechanism part, we can see this semicircle thing. This is the polypeptide chain, and this is the active site of ribonuclease A, and the shaded portion is RNA, that is the substrate. So this is a substrate and this is the active site of the enzyme, and you have histidine 12 here, which is a deprotonated form which can act as base, and histidine 119 here, that is protonated form of histidine, this can act as acid. And the first step, histidine 12, which is a base which abstracts a proton from the 2' OH of RNA. Now the protons goes here, and now the 2' OH becomes O minus, and this O minus nucleophilically attacks phosphorus and forms a PO bond. At the same time, histidine 119, which donates a proton which helps in the cleavage of this PO bond, and this proton goes here and forming CH2OH, and this portion of RNA is released. Now you are getting 2' 3' cyclic nucleotide here, and histidine 12 is in the protonated form and histidine 119 in the deprotonated form. And this 2' 3' cyclic nucleotide, which is the transition state, and that is stabilized by lysine 41 by interacting with the phosphate group. And the next step of the reaction is hydrolysis of 2' 3' cyclic nucleotide by water. So this is the second step of the reaction. Now histidine 12 is protonated and histidine 119 is deprotonated, and this histidine 119, that is the deprotonated form which acts as a base, this abstracts proton from water, and now this OH, which O of the OH nucleophilically attacks phosphorus and it is forming a POH bond. Same time histidine 12, which is protonated form, that acts as acid, donates proton, which helps in the cleavage of this PO bond, and here 2' OH is formed and this portion is released. Now you are getting histidine 12 in the deprotonated form and histidine 119 in the protonated form. This is the second phase of the reaction, and overall reaction is depicted here. First phase and second phase combined together. In the first phase, histidine 12 acts as a base, abstracts proton from 2' OH. Now this O minus nucleophilically attacks phosphorus. It is forming a PO bond. At the same time, histidine 119, which acts as acid, donates proton, and this proton helps in the cleavage of this PO bond, and this proton goes with this CH2O and forming CH2OH, and this portion is released. Here the 2' 3' cyclic nucleotide is formed and histidine 12 is in the protonated form and histidine 119 is in the deprotonated form, and this cyclic nucleotide form is intermediate, which is stabilized by lysine 41, and the second step is hydrolysis of this 2' 3' cyclic nucleotide, and that involves water molecule. And the second step, the histidine 119, which is acting as a base, abstracts a proton from water, and now this O minus H, that is this O minus of OH attacks phosphorus nucleophilically, forms POH bond. Same time histidine 12 donates its proton, which comes here, helps in the cleavage of this bond, and it forms HOH bond at the 2' position, and this portion is released. And the enzyme is coming back to the original condition with the histidine 12 in the deprotonated form and histidine 119 in the protonated form. This is the overall reaction, the simplest mechanism, which is followed by ribonuclease A, that is a concerted general acid base catalysis. Hope this is clear to you. This time also I have few questions for you. 1. Which reaction is catalyzed by RNase A? 2. Name the active site residues of RNase A. 3. Which histidine is acting as acid in the first phase of RNase reaction? 4. Name the intermediate formed during RNase A reaction. 5. What is the role of lysine 41 in RNase catalysis? Answer these questions and submit it in Google Classroom. If you have any doubt, please let me know. Thank you. Stay home, stay safe.